Welcome back to Quick and Dirty Med School. Tonight we'll be covering the cell signaling pathways. There are two major types. The first is intracellular, and that occurs within the cell. It's responsible for rapid responses, and it is important for the moment-to-moment regulation of metabolism. It affects the availability of substrates, product inhibition, and altering the allosteric activators or inhibitors. Intercellular signaling occurs between the cell. There is a slow response, but the signal lasts longer than intracellular signals. They are a long-range integration of metabolism, and it works through these agents, hormones, and neurotransmitters. Chemical messengers are secreted from one cell in response to a specific stimulus. They travel to target cells and they bind to specific receptors. These are the examples. So an endocrine signal is a chemical signal that will be distributed in the blood, and those bind to distant targets. Paracrine are a local action by diffusing from the source cell to nearby targets. And autocrine are chemical signals that act on the cell that made it. Let's go a little bit more in depth. Intracrine, the cell will create the signal, and that will only work within the cell itself. Autocrine, the cell creates the signaling proteins, but it has to be secreted out. It crosses the cell membrane and binds and activates the receptor on the outside of the cell. Paracrine, let's just say it was secreted from the cell, and once secreted, it crosses the membrane and then goes into the local circulation. And the local circulation will, provide the transport to only nearby cells. So this works within only like a small clump of cell. Endocrine is when the cell secretes the signal into the general circulation, and it will be transported until the signal reaches its distant target cell. And then last are the neuroendocrine, and that is only involved in the axons and the synaptic cleft. And then once those are released into the general circulation, the signals will go to the distant target cells. So on the receptor, there are specific binding sites, and once bound, they will induce conformational changes. And they can interact with proteins or DNA. Plasma membrane receptors span the membrane, and they have an extracellular binding domain. The signal transduction is the route where an extracellular signal will bind to the receptor, and then the receptor will bring the signal in and then convert it into an intracellular response. Plasma receptors are found on the outside of the cell. They are hydrophilic. And intracellular receptors are going to be hydrophobic, so they're going to be within the cell. So there's these different types of pathways, which we will go through next. But plasma receptors are heptahelical receptors. They have 7 alpha helices spanning the membrane. And these are the examples: the G protein coupled receptors, and the kinases, or the ion channel receptors. Right. So hormones can provide the signal for a positive or a negative response, even within the same tissue. And the response really depends on the number of receptors occupied. Lipophilic are hydrophobic, and they are steroids, and hydrophilic are the peptide hormones. And peptide hormones can only stay on the outside of the cells, and they have to be brought in via the second messenger process. Lipophilic hormones can diffuse the lipid bilayer and the plasma membrane to interact with the intracellular receptors. They do require the transport proteins They have a longer half-life, and they're gene-specific. They can bind to the transcription factors, which bind to the enhancer binding site on the DNA. Examples include the steroids, vitamin D, and vitamin A, which is retinoic acid. So here's the superfamily. We should know this. So the zona fasciculata will be involved with cortisol and corticosterone. Zona glomerulosa will be involved in aldosterone. And then the zona reticularis is going to be involved in androstenediones. Ooh, hard to say. But we can know this because fasciculata is involved with the muscles and cortisol. So cortisol is a stress hormone. Zona glomerulosa, that's the glomeruli, and that's going to be involved with the kidneys, such as aldosterone. And the reticulus will be involved with more of like the sex hormones. Hydrophilic hormones can only interact with the plasma membrane-bound receptors. They don't need the transport proteins, and they have the shorter half-lives, and they are peptide hormones. All right, let's go into the signaling transduction. The heptahelical receptors are G protein coupled receptors. Those are the most common plasma receptors. They work via the secondary messenger system, and that amplifies the strength of the signal. So once bound to the plasma receptor, it makes the signal stronger when it is starting to be invaginated or brought into the cell, and then it is transported to the nucleus, and then it is bound, and then once bound it can enhance a gene, which will also amplify the strength of that signal. So these are the different types. And the adenylated cyclase system is the G alpha S system, and that is when a ligand binds to the surface receptor and induces the conformational change, which will trigger the transmembrane signal. So once bound, here we go, the activation of the G proteins will activate adenylate cyclase. And then the G protein in particular is the GSA, and that will use an ATP and attach to the adenylate cyclase, and then the ATP will be converted into a CAMP, and the CAMP will release protein kinase A, which will attach to something else, such as various catalytic subunits that will go into the nucleus, and then affect gene transcription and expression. There we go. So in a similar pathway, a hormone will bind to an inhibitory receptor, and then the G proteins will be activated, but the GI receptor will bind to the adenylate cyclase, and it will inactivate it. So no CAMP is made. However, the protein kinase A, pKa, will be made, and that will also play a role in phosphorylating other catalytic subunits. But those catalytic subunits will not play a role in it will not play a role in increasing the expression of the gene. It will be used to inactivate. So another one is the GAQ. That dissociates when the ligands bind to the GPCR subunit. and that interacts with the phospholipase C, which will cleave PIP2 into IP3 and DAG. IP3 is the water-soluble version, and it will diffuse into the endoplasmic reticular. DAG will stay on the outside of the cell, on the cellular membrane, and once IP3 is attached to the endoplasmic reticulum, it stimulates the release of calcium ions. The more calcium ions there are, it stimulates calmodulin, which will activate other protein kinases, which in turn activate the calcium binding kinase, and those will phosphorylate the regulatory proteins, receptors, and transcription factors. So tyrosine kinase is a tyrosine-specific enzyme, and the receptors contain an extracellular domain for the binding of the ligands and an intracellular domain with tyrosine kinase activity. So the interaction of the external domain upregulates tyrosine kinase. So ligand comes, binds, and the receptor is dimerized, and tyrosine kinase is activated, and then it will it will cause the phosphorylation of the tyrosines and cytoplasmic tails. So phosphorylated tyrosines are recognized by adapter proteins, and those activate other proteins when bound, and the receptor tyrosine kinase is turned off by the tyrosine phosphatases by dephosphorylation. As you can see by these long and lengthy pathways, these are all secondary messengers. So these cell signals that I have been describing don't work on the inactivation or activation like directly. They have to be passed, then phosphorylated, and then bound to the binding site in DNA in order for them to express or depress a gene. So this is the insulin JAK-STAT pathway. These are the non-tyrosine kinase receptors, and once a cytokine is bound to the Janus kinase, it will be the JAK receptor will be dimerized, and that phosphorylates the tyrosine residues of the STAT proteins, and then those will enter the nucleus, and they will bind to the DNA sequences in the promoter sequence, and then through translation, it will create insulin. This is a relatively rapid response. Insulin levels rise about an hour after you eat, but that is pretty fast for a secondary pathway. So the genetic aberrations that involve the JAK pathway are polycythemia vera, essential thrombocythemia, a primary myelofibrosis, and then the progression of tumors. So cytokines are secreted from the leukocytes. They stimulate humoral and cellular immune responses. Lymphokines are made from the lymphocytes, and monokines are made from the monocytes or the macrophages. Interleukins will affect the cellular response of the leukocytes, and growth factors that target cells of hematopoietic origin. So this is the path of the serine threonine kinase. So the TGF-beta will bind, and then it will be phosphorylated, and then it will bind to another kind. This will be it will bind to a type 1, and then the phosphorylated type 1 recruits and phosphorylated the SMAD proteins, and then that regulates osteogenesis and developmental processes. So ion channels are involved in, the synapse, and neurotransmitters will come, bind, cause a rapid opening of the ion channels, and then a signal will be passed down, and those can be used either to promote or inhibit. Right, steroids, since they are, hydrophobic and lipophilic, they diffuse directly across the plasma membrane, through the cytosol, and right into the nucleus. Those can bind and cause a receptor homodimerization, and those homodimers will move into the nucleus and bind to the hormone response element after recruiting the HistoneAcetylases. Remember when we were talking about transcription, translation a while ago? Histone plays a role in all of that by unwinding the DNA enough for the signal to come in and bind. So nitric oxide synthase is responsible for the creation of nitric oxide, and inducible nitric oxide synthase is made in the macrophages, while endothelial nitric oxide synthase is made in the endothelial walls or cells. Both types make a nitric oxide from arginine with oxygen and NADPH, and then acetylcholine will bind to the endothelial cell, and nitric oxide diffuses locally, causing vasodilatation. So then it will bind to the guanylyl cyclase and make a cGMP, which will relax the smooth muscle, and that will cause the vasodilation. So it's used in myocardial infarction or heart attacks. And you will give the patient isosorbide mononitrate sublingually, so under the tongue, and then by that metabolism in the muscle, it will create the vasodilation, and it is a sulfhydryl dependent mechanism. That's a little bit intense for us, so I don't think we need to go very far into it, but remember, isosorbide mononitrate for vasodilation for the myocardial infarction. So cGMP is involved with nitric oxide. So once nitric oxide is able to cross the membrane and become soluble guanylate cyclase with the help of nitric oxide synthase, it will create the cGMP, and the cGMP specific phosphodiesterase type 5 will accept it and break it down to the GTP, or it will go into the protein kinase G and then go to the smooth muscle as myosin phosphatase for relaxation, or VASP for the platelet inhibition, or the transcription faction that will change gene expression. So these are found in the corpus cavernosum and the retina, and they inhibit specific isozymes without side effects. So this is actually a hot topic for research right now. It is used to promote vasodilation in these drugs, Viagra, Levitra, and Cialis, and they are used to treat erectile dysfunction. So these are the sites of signal termination. So stimulus occurs, the terminator is released, and the messenger will diffuse or degrade, and then once bound to the receptor, the desensitization down regulation will occur by dephosphorylating products, and GTPases will help the signal transduce into the second messenger system, and then the phosphodiesterases will also help with the termination response. And then once the response has occurred, the expression will be ceased. So thank you for your attention and good luck studying.